Good day, my name is Sonora Meiling, and today I'm going to be discussing the first quantitative relationship between stony coral tissue loss disease and temperature, in which thermal stress abates lesion progression. Stony coral tissue loss disease is categorized as a rapid tissue loss disease and is very visually distinct. In this photograph, there's a large infected colony of Colpophyllia natans with two white blotches. These are the area in which the tissue has necrosed and the underlying skeleton is exposed. The lesions appear stark white because they have progressed so rapidly that algae has yet had the time to recruit to the bare substrate. But what makes this tissue loss disease so unique compared to others is just how quickly these lesions progress. As you can see in this approximately weekly photo progression, those two lesions rapidly expand, coalesce, until the colony experiences full mortality in just over a month. However, this disease doesn't affect all species of sclerotian corals equally. In this diagram, susceptibility is differentiated into hierarchical categories distinguished by the typical order of infection. On the left are the highly susceptible species, in which we anticipate showing signs of disease first, followed by the intermediately susceptible species, which includes the brain corals, and the moderately susceptible species, which includes the boulder and mounding corals. Stony coral tissue loss disease was first observed in the United States Virgin Islands at the end of January in 2019. Initial documentation came from the red star on the south side of St. Thomas, which denotes Flat Cay reef. Since then, aggressive monitoring has mapped the geographic expansion of this disease throughout the territory. The light pink area of the polygon depicts the disease boundary, or the areas of new observation of disease. And the salmon area of the polygon depicts the total area of infected reefs. And after about a year and a half, you can see that the disease has encompassed almost the entire island of St. Thomas, including both near-shore shallow reefs and offshore mesophotic reef systems. The disease has also spread to the neighboring island of St. John, and most recently, the British Virgin Islands. However, when this disease was first observed in January of 2019, we weren't confident it was the same thing that was being documented in Florida, which led to multiple collaborative studies and experiments, one of which I'll be talking about today. For the present study, I compared the lesion progression rates of multiple species infected with what we believe to be stony coral tissue loss disease at the yellow star, which is locally known as Black Point Reef. To do this, I analyzed 3D models of different infected colonies. I chose to use 3D photogrammetry because of its extremely high resolution. On the left, there's a photograph of an infected colony of Colpophyllia natans, and on the right is a screenshot of the 3D model constructed of that same colony. And as you can see by the almost indistinguishable detail between the two images, these 3D models have high credibility. To build these models, divers visited marked coral colonies once a week, every week for 6 months. An approximately 2-minute video was recorded of each coral colony, ensuring to move slowly and steadily, circumnavigating the entire colony and covering the full surface area. From these videos, I extracted the highest quality frame from every second and imported them into the program Agisoft Metashape for 3D reconstruction. The first step is building a sparse point cloud in which the software finds every overlapping point of the images and aligns them into 3-dimensional space. The second step is to build a dense point cloud in which the software uses those overlapping images and depth maps created from them to create more points into 3-dimensional space. However, what you're seeing here is millions and millions of points in which we can't measure a surface area of, which leads us to the next step of building a cohesive mesh, and finally overlaying a texture from those overlapping images on top of the mesh to create the full 3D model. Once the reconstruction is complete, I applied a scale to the models so that measurements could be made in real space. These measurements were conducted in the program Cloud Compare, and for this study, I chose to use the change in healthy tissue area as a proxy for lesion progression rate because it's easier to distinguish the boundary between the healthy tissue area and the surrounding substrate than it is from the old mortality and the surrounding substrate. So the first step is to remove the surrounding substrate, the area of old mortality, recent mortality, leaving me with just the area of healthy tissue. This was repeated for every coral colony at every time point for 5 species, resulting in just under 400 models for analyses. Using repeated measures ANOVAs, I ultimately found that the lesion progression rate decreases over time. In the figure on the right, the x-axis depicts the different periods of monitoring, and the y-axis depicts the aerial lesion progression rate in centimeters squared per day. As you can see with the white dashed line, there's a significant decrease in the lesion progression rate over time. And then, as you can see with the overlap of the full colored lines, which show the different species monitored for this study, there's no difference in the lesion progression rate among species, which ultimately suggests that once infected, stony coral tissue loss disease affects all susceptible species equally. I then analyzed the proportional lesion progression rates for this time period in all species of monitoring. To calculate these values, I divided the change in tissue area by the previous area of healthy tissue and found that there was no difference in the proportional lesion progression rate over time nor among species, which suggests that the lesion progression rate is consistent throughout the duration of infection. And as I mentioned earlier, we weren't confident this disease was the same as was being documented in Florida, so I wanted to compare the rates that were being reported here to what was being reported in Florida. In this table, the far left column depicts the different species with reported lesion progression rates, and the sequential columns are different studies. Lesion progression rates reported in green are significantly lower than those found in this study. Those reported in red are significantly higher than those reported in this study. And those reported in black are approximately equal to those reported in this study. And as you can see, the lesion progression rates between this study, which is in the field in the USVI, and the study by my advisor and I from a lab-based transmission experiment, there isn't a consistent increase or decrease in the lesion progression rate between the two studies. And once the lesion progression rates are compared to Florida, there's a significant increase. But this doesn't necessarily mean that we're observing a different disease here than is being documented in Florida, nor that the corals in Florida are necessarily dying faster than those in the USVI, because the rates reported by Abby and colleagues in Florida are approximately equal to those in this study. And although there are differences between the different studies, regions, and species in the lesion progression rates, I want to put into perspective just how rapid these corals are dying. So the final column shows the aerial lesion progression rate for white plague, which was previously one of the most threatening rapid tissue loss diseases in the Caribbean. And as you can see, it's a magnitude lower than even the slowest lesion progression rates reported for stony coral tissue loss disease, which really emphasizes just how devastating this disease is and is going to continue being for reef communities in the Caribbean. And if you remember, I monitored corals for a full duration of 6 months, and the first 2 analyses were only on the first month of monitoring. And that's because too many of the colonies of the 3 other species died after that first month of monitoring for statistical analyses. So for further analyses, I will only include the 2 species, Montastraea cavernosa in gray and Orbicella annularis in orange. Axes are all the same for this figure as the previous, and you can see that there's a significant decrease in lesion progression rate over time still as we look at the full duration of monitoring. There's also a significant difference in the lesion progression rate between the 2 species. However, some interesting patterns arise whenever you look at the individual replicates within these 2 species. Multiple Montastraea cavernosa colonies, lesion progression rates significantly slowed down at the beginning of October. And ultimately, all of the lesions on the Orbicella annularis colony stopped by the beginning of October. which both of these trends coincide with the onset of thermal stress, which ultimately resulted in the second largest bleaching event this territory has experienced in the past 2 decades. The x-axis of this figure depicts 1 year of time from January 2019 to January of 2020. The top portion of this figure shows the specific temperature at Black Point Reef in degrees Celsius, and the blue line is the bleaching threshold specific to Black Point, calculated by Smith et al. In 2016. The bottom portion of this figure depicts the degree heating weeks, which are just a proxy for accumulated thermal stress. In the black box is the time period for monitoring during this study, which shows that I have lesion progression rates both before, during, and after this mass bleaching event. And what's interesting is that on October 14th, which is the initial peak of thermal stress at 9.13 degree heating weeks, 50% of the lesions had stopped on the surviving colonies. And what I further found is that there's a stronger correlation between the lesion progression rate and degree heating weeks than there is between the lesion progression rate and temperature, suggesting that the abation of these lesions is actually related to the thermal stress experienced by the coral and their algal symbionts, rather than a manipulation of the virulence of this disease due to the elevated temperatures. We also analyzed the proportional lesion progression rates for these 2 species for the full duration of the monitoring, and found that there was no difference in the proportional lesion progression rates over time, nor between the 2 species, suggesting that the lesion progression rate, again, is consistent throughout the duration of the infection, which doesn't match what we were just looking at with the absolute lesion progression rates. But whenever you look at the individual colonies within the 2 species, you can see a little bit more of the pattern of the Orbicella annularis colonies stopping at the onset of thermal stress, and a couple of the Montastraea cavernosa colonies slowing down and stopping during thermal stress. However, there's a lot of variation in the proportional lesion progression for the Montastraea cavernosa, suggesting either variation within that species in their resilience to the disease, which maybe a larger sample size would have been able to detect this relationship between the proportional lesion progression rate and degree heating weeks or thermal stress. Which ultimately raises the question, does stony coral tissue loss disease pathogen or pathogen infect the endosymbionts? Potentially, which might line up with some of the research released by Landsberg and colleagues in 2018. In this image of a histopathology cross-section of an infected polyp of Montastraea cavernosa, they found many fuchsia dots within the gastrodermis, which are crystalline inclusion bodies, which are indicative of a viral presence. Many of these crystalline inclusion bodies are found within the vacuoles containing the endosymbiont algae, or zooxanthellae, which provide the coral with their energy, which helps support that notion that stony coral tissue loss disease pathogen or pathogens has some kind of relationship with the endosymbionts. And one might think, considering many studies have shown corals recruiting more thermally tolerant clades after bleaching, and the colonies in this study that were previously infected then bleached didn't develop any new lesions, that specifically the stony coral tissue loss disease targets the less thermally tolerant clades. In conclusion, based on the wide range of species affected, extensive geographic range, and rapid lesion progression, stony coral tissue loss disease already has and is going to continue having the largest impact to Caribbean reefs documented to date. Through the analysis of 3D models and intensive monitoring, I have shown that lesion progression rates abate at the onset of thermal stress. However, the proportional lesion progression rates are consistent through time, which the relationship between the thermal stress and the absolute lesion progression rates can ultimately be used to the advantage of resource managers. During these bleaching events, when lesions stop on some colonies, there's a natural filtering of the resilient genotypes that would be beneficial for restoration. And while intervention should not only be applied during and following bleaching events, I strongly suggest efforts are increased during this time, because there will be reduced disease prevalence on the reefs and thus resources can be more effectively distributed. I'd like to give a huge thank you to my funding sources via EPSCoR, NSF Rapid, Akovi Karakus, and the Lonaventa Foundation. I'd also like to give a thank you to my master's thesis committee, supporting lab and technicians that helped me with the field and technical portion of this project. For more information, you can visit vicoraldisease.org for general information about stony coral tissue loss disease in the territory and what we're doing to combat it. But for specific information about this study, you can email me at the email posted, and I look forward to your discussions. Thank you.